The overall goal of this procedure is to culture bacteria in the system for analysis at the liquid vacuum interface, or SALVI, for subsequent imaging of the biofilms in their natural hydrated state with vacuum-based techniques such as time-of-flight secondary ion mass spectrometry.
We present a method that helps answer key questions in environmental microbiology by visualizing the chemical spatial distribution of living biofilms using imaging mass spectrometry like SIMS. The main advantage of this technique is that we can introduce biofilms alive to a mass spectrometer and many other analytical platforms to obtain multiplex measurements.
Although Shewanella oneidensis is used in this video, this method can also be applied to anaerobic chambers for biofilm growth. The latter requires a more stringent growth environment.
Visual demonstration of this method is critical as the tubing setup steps are difficult to learn. Specific placement of tubing and care are necessary when transferring to the biosafety cabinet.
Using a 22-gauge needle, carefully punch a hole through a serum bottle rubber stopper. Cut 20 inches of 1/32 inch PTFE tubing with a razor such that one end of the tubing is pointed. Using the pointed end, thread the PTFE tubing through the hole in the top portion of the rubber stopper. Push the tube through until roughly 2 centimeters is through the stopper and cut the pointed end of the PTFE tube with a razor to have a flat end. Remove the plunger of a 5 milliliter syringe and fit the rubber stopper into the open end of the syringe. The 2 centimeter end of PTFE tubing is within the barrel of the syringe. This is the bubble trap. Wrap the bubble trap with aluminum foil and secure it with autoclave tape.